All right, everybody, welcome back. In our last lecture, we were examining how DNA is structured within the cell. And we specifically were looking how DNA are wrapped around these histones and then are stored in these tight units called chromosomes. And now we're going to sort of be examining chromosomes a little bit more specifically. So when we're talking about human DNA, we're talking about 23 pairs of chromosomes, where one chromosome pair is inherited from the biological mother, and one chromosome is inherited from the father to create each individual chromosome. So you can see here, this is how we sort of line up chromosome. Oh, sorry. Oh no. Going back. Ugh, I lost my pointer. Ah, there it is. Okay. So when we look at DNA sort of laid out this way, we can see all 23 pairs of chromosomes organized. So you get one chromosome from your father, one chromosome from your mother. And lastly, this 23rd pair, this is what determines quote unquote biological sex, where one is the X chromosome, which is inherited from your mother, and one Y chromosome. And essentially, when we're talking about biological sex, we're really talking about the organizations of these two chromosomes. So when we're talking about biological sex, again, this is only biological sex, nothing more. A typical biologically sexed female would have an XX chromosome, and a biologically sexed male would be an XY. However, it's important to note that there are many variations that occur in these chromosomes. And I put in your additional videos for this week some interesting studies looking at both the biology of gender, which is, again, very different than sex, intersex, which is understanding variations that occur in terms of different sexual traits. For example, in Klinefelter syndrome, you can actually have an additional X chromosome, so you have an XXY, or you could have only a single X chromosome. And there's lots of variations that can happen, that can occur in here. And in intersex individuals, you can have even more variation where there is differentiation in both gonads and in different hormone levels, which can have dramatic changes in the body. And I put in a few additional lectures that sort of look specifically at this phenomenon, and I think they really do a great job of sort of understanding the differences that can actually occur, because they're myriad. And it's important to spend some time understanding how biologists understand, again, this concept of biological sex, where we're really only interested in understanding an X and a Y chromosome, to what actual functional biology can actually look like, which can be very different. The main takeaway I would like you to understand here is that while there are X and Y chromosomes, there's a huge diversity of what actually can happen in biology, and that's something to be celebrated. So while they may say something about sex, not always necessarily, they definitely don't have anything to say about gender. And there are very interesting studies right now that are really understanding the whole diversity of sexual characteristics in biology. And I hope to sort of showed some good examples in the extra videos for this course this week. So when we're talking about structure of DNA, so again, I want you to sort of think back to those macromolecules of life that we talked about, and sort of start to see similarities and differences that you see. We're not a chemistry course here, so we're not going to go too much into this. But it's good to start to understand and draw connections between, again, those macromolecules of life, and how they're broken down into these very important subunits that then may produce and be an important part of the structure of DNA. So the first thing that's really important is this nitrogenous base area, followed by this 5-carbon sugar, and then this phosphate group. So again, based on what you know about macromolecules, what types of macromolecules would help generate structures like this? So you've probably, when you've studied DNA in the past before, you've probably come across sort of fancy pictures that look like this, that show that we have this phosphate is become sort of the bands, the structural bands on the outside, and then this sugar base. So we have this sugar phosphate group that's holding the sides of the DNA, and then they're joined together by this base group. And this base group varies, and there are four specific base groups: adenine, thymine, guanine, and cytosine. But rather than spend too much time just sort of looking at a glossy picture like this, what again do those structures actually look like? So when we're talking about the physical structure, so again, this is not a chemistry course, so don't freak out too much when you see a big image like this. Let's actually break it down. So first, let's identify what is the outside of the DNA, and it's these phosphates, right? So we can look at these phosphate bonds, and we see that the phosphate binds to this sugar. Okay, so we have phosphates binding to sugars, and this is what is creating that backbone of the DNA. And then we have these base pairs where we have, in this case, thymine, adenine, guanine, and cytosine. And again, in your previous biology courses, you probably memorized that, okay, cytosine always pairs with guanine, thymine always pairs with adenine. But let's look at why exactly that is. And again, it has to do with this chemical structure. So you can see that there are actually, with cytosine bonds, there are actually 3 points of nitrogen bonding that happens here, where with thymine and adenine, there are only 2. So an adenine can't actually bind to a cytosine because, again, these binding specific points fit together essentially like puzzle pieces. And that's why adenine and thymine always go together, and guanine and cytosine always go together. So let's zoom out again and sort of understand what that looks like. So the 2 strands of nucleotides pair up and they twist around to each other to create this spiral double helix shape that we're very familiar with. So in our previous slide, we were only looking at a section, in this case, this 4-base section where we have adenine, thymine, cytosine, and guanine all together, except I guess it would be inverted in this case. So now we can see sort of, again, trying to make a longer strand of DNA. And so we have these phosphate bonds make up the outside, so that's this blue. And you can see that it becomes this blue in this classic shape. And then we have the different bonds of those base pairs, the adenine, thymine, guanine, cytosine. And that's what creates this shape, which then, in turn, twists around those histones, and those histones bind together to create those dense chromosomes that we saw and talked about in our previous slide. And I think that's where we'll end today, is just understanding, again, this distinct structure of DNA and understanding chemically a little bit of what's going on to actually create this classic double helix shape that you're probably also familiar with. Okay, we'll pick up soon. See you soon.